the role of grass, silica, phytoliths, and roots in soil health. So this is a joint research project between the K-State Department of Agronomy in collaboration to study the role of grass, silica, phytoliths in soil health, and also the Brazilian Sea Control Light Source and the Canadian Light Source, where we have projects going. This collaboration is to build a multidisciplinary research team to develop imaging techniques to analyze phytoliths chemistry and physical structure at nanoscale never recorded before. So most of you, I'm not sure if you are familiar with phytoliths. So phytoliths are unique structures made by plants. They're very beautiful microstructures made by silica, made of silica, and for many plant species. They are found in many sizes and shapes. In here, this picture, you can see many shapes and sizes of these silica bodies made by plants. And we can find them in many plant species in a whole plant. We can find phytoliths in the roots, in the stem, leaves, and reproductive organs, and also in the grains. So it is, phytoliths are very used in archaeology and related studies because they last for centuries in the soil or even more, and people can use them as proxies to reconstruct the past. For example, the past vegetation, past human activity in agriculture and related fields. So they also have very unique chemistry. As I said, they are made of silica, but while they are made by plants, certain types of inorganics can be incorporated. In this image here, we can see some little piles of silica, which is absorbed from soil and transported to the within the certain types of cells. And also we can find inside phytoliths organic carbon and nitrogen. This organic carbon is part of this project between synchrotron light sources, where we are trying to figure out where the carbon is located and the quantity, but I'm not going into details today. And the phytoliths chemistry will depends on soil type, plant species, and rarity, and also climate. For example, it depends from precipitation. For example, if it's very dry, the plant can absorb more or less silica. So phytoliths, as I said, they last for centuries in the soil, so they're very rigid microstructures, and then they play several functions in the plants. They provide mechanical strength. For example, they help plants to be stand after like wind gusts, and they can be more erected for light absorption. They protect plants against stresses, abiotic like drought, UV radiation, and against biotic stresses, for example, against fungi, nematodes, and herbivory. For example, this is a grasshopper. I think most of you know them, and then they love grasses, right? And this is their native mandible, which is like more or less like a normal teeth. But when they are fed by high silicon content grass, in this case rescue grass, their mandible can be get damaged by feeding high silicon content plants. So they don't last too much if they eat high silicon content plant. And phytoliths are, as I said, they are silicon deposits in the plants, and then we can find them in many plants we use to in many crops we use to for food and energy production. We can find phytoliths in wheat, barley, rye, rice, maize, oats, and millet. And that's why they are very ubiquitous in soil. So we can find phytoliths in everywhere, even in forest and savanna and grasslands. We can find in different soil profiles as well. Here I bring an example of silicon in food crop plants. We can find plants and grains with high silicon content. For example, in whole grain bread, we can find more or less 2.3% of silicon. Wheat bran is very rich in this inorganic element. And soybean as well, it depends on the soil variety. And the quantity of phytoliths in it also, as I said, is also changed according to the plant organ and plant species. For example, in this table, there are many examples, and I want to highlight here wheat. We can find it in leaves and stem in concentrations that varies between almost 3% to almost 8%. This is a study from wheat varieties in Europe, but I found some data in here in the US. It's more or less about 5% of silicon. For sorghum, we can also find in the leaves about 4%, and the roots, as I said that image before, and also in corn. It depends the type of soil, of course, and we can reach a very high concentration of silicon. And also, as I said, for energy plants like sugarcane in miscanthus, we can find a good silicon concentration. For example, this project is started in Brazil. So in São Paulo state, we use sugarcane to produce sugar and ethanol, and we have plenty of sugarcane biomass to burn and produce energy. So leaves and stems are rich in silica, and that's why I'm gonna talk a little bit more about sugarcane. And miscanthus is important for example, and for here, we use them for energy production as well. And the shoots will have more or less up to 4% of silicon, but if we work with biochar, for example, it can reach up to 40% of silicon in the material. So as I said, they are used for energy. Mostly they are burned to produce energy power, at least in Brazil. And the main residue left after burning this type of biomass is ash. So we would like to see, to study if ashes focusing on silica content will change some soil properties. So we developed a microcosm experiment. Is a proof of concept actually, where we designed to study the role of phytoliths and roots in the soil community and soil aggregation. To isolate phytoliths, we also burn the biomass. This is sugarcane epidermis internode, where we can find these little guys here are phytoliths. They keep quite well after burning this material, about at roughly 600 Celsius degrees. So they last for at high temperature. And this material I got from sugarcane internode epidermis. I use only the epidermis, which is rich in silica, phytoliths, and the carbon nitrogen ratio, it's about 57. And also I used here miscanthus shoot from North Farm. It's an ongoing experiment, but I use the same method to obtain the ashes. So I burn it, but I digest it in an acidic solution, and then I burn it again. It's more or less 600 Celsius degrees to get a material rich in silica. And then here I have a material rich in carbon and silica. So they are quite a little bit different. And then also you can see their shape are more or less similar, because this is a very common phytolith from grasses. It's a way to define those species as well. So the microcosm proof of concept, we used plant ashes from miscanthus or sugarcane and silt clay loam soil, and then we sieve it. We use sieve with 53 micron mesh opening, and we put it inside frames where it actually is a 3D printed frame. We can remove this part here, and then we can see the roots and sample it in these dimensions. We printed out these frames in here at K-State. That's why they're also purple. And we designed it for this experiment with 6 treatments, where I use only soil and plant, soil and ashes mixed in the whole soil. And then I used local ash to see if the plant will respond differently to mixing or not. And I used here mixed ash with plant, and mixed ash and plant, and a commercial inoculum of arbuscular mycorrhizal fungi. And then this is how the microcosm looks like. The plants with switchgrass can grow quite well in this experiment. We can get many things from here. And then here is an image of the shoot and root development over 10 weeks. This is in the very beginning. We can see the microcosm, the switchgrass growing little plantlet. And then here the ashes, as I said, located in one single spot. And here after 10 weeks, here I cut the plant, and what remained was the roots. We can see the main root here growing in this direction. And here, when one of the purpose of this microcosm is to see the root development, and we can measure. We can use this in the future studies to measure this roots and the whole the whole structure. This is how we took the soil out. So very gentle, we can remove soil from roots, and then you can see here it keeps the whole structure, and then we can study like little spots from the roots. And here I bring some results about the soil microbial community. And from the 6 treatments, we analyze the phospholipid fatty acids, which gives to us idea of how the microbial community responds to to the treatments. So for microbial biomass, actinomycete and fungi and gram-positive bacteria, we didn't see any difference. But in here for gram-negative bacteria, we saw an increase in the activity from gram-negative bacteria in the treatment with ash mixed. And for AMF, which is arbuscular mycorrhizal fungi, we saw one statistical difference in here in this treatment with plant and ash mixed. And also we analyzed the mycorrhizal colonization. We didn't have any statistical difference, but we can see here we have a little bit more root colonization of mycorrhizal fungi in comparison to the other treatments. And about soils fertility and for the 6 treatments as well, we analyzed some micronutrients like phosphorus, zinc, iron, copper, and manganese. And for phosphorus, we can see here in the dotted boxes, when we apply ashes, we are also applying certain elements, some nutrients. In this case, for phosphorus, we can see high concentration of phosphorus in comparison to the bulk soil when we add ashes. In this treatment here, we can see the plant used a little bit of this phosphorus for its growth, but here we don't see any difference because we use AMF. It might have some effect here in the plant not using phosphorus for at least after 10 weeks. For manganese as well, we see we have a little bit increase of this element in the treatments, and also after we plant growth. And for cation exchange capacity, we didn't see any difference. And for elemental analysis for carbon and nitrogen, we can see in comparison to the bulk soil, when we apply this ashes, sugarcane ash, which is rich in also in carbon, we keep this carbon preserved in the soil, and the carbon keeps more or less the same in between the treatments, but the nitrogen is used for microorganisms or the plant. It is hard to say. And also for the CN ratio, we can see a good effect of applying sugarcane ashes in the soil. It seems like we keep the soil, this carbon preserved, which it could be very interesting for soil carbon sequestration strategies. And for plant biomass, what we saw was if I apply the ashes in a local spot, the plant can use it more efficiently. So the response was more shoot biomass in this treatment. And we also took some images of soil aggregates. After 10 weeks, we didn't see too much aggregation, but in some scanning electron microscopy, we can see here, for example, one phytolith. It might serve after a while. It may serve as a seed for aggregation, but also we see effects of root hairs and hyphae. I don't know if you can see here, but there are some kind of cords. They are fungal hyphae, which also helps for to keep those soil aggregates together. And I would like to thank you to FAPESP, which is the agency that funds my project. Here at K-State and small, the Soil Microbial Agroecology Lab, Valent, and USDA for providing the seeds. I'm open for any questions. Thank you.